Um, Good morning. Welcome to today's science lesson, and we are going to touch on the unicellular and multicellular organism. As we had seen earlier, we had said that unicellular organisms are organisms that are made up of a single cell. And that's why we call them unicellular organisms. Then with multicellular organisms are organisms that are made up of many cells. They are made up of many cells, and that's why we call them multicellular organisms. We can start by looking at an example of plants. What are plants and animals are big organisms. And because they are large organisms, they are multicellular. They are multicellular, and because of that, they are made up of billions of cells, and all adding up together, billions of cells, they are made up of billions of cells together. And they help, these billions of cells, they help the plants and animals to survive. Those tiny unicellular organisms, we cannot see them with our naked eyes. We need to use a microscope. Therefore, we say many microscopic organisms are single-celled organisms. They are unicellular. And example of these microorganisms that are unicellular, that we can use microscope to see them. Examples we have are the bacteria. We had said that the cyanobacteria, they are also unicellular. And some of the protists are also unicellular. We are saying from the protista kingdom, we have protista are organisms that have nucleus, a nucleus, and they live independent. They live as independent cells in colonies. They are not differentiated. They don't have tissues. There are about 85,000 unicellular organisms, but they are yet to discover other close to 1 million unicellular organisms that we are yet to study. Many unicellular organisms are plant-like. They operate, they're either plants, they look as if they are plants that have some characteristics which try to depict them as if they're plants, while also others depict some characteristics as if they are animal-like. Then we are saying some of the plant-like unicellular organisms, they have their cells have a cell wall. And because they have a cell wall that is made up of cellulose, they are able to make their own food. The unicellular organisms which have a cell wall made up of cellulose, they are able to make their own food. We have other organisms that are unicellular, and these organisms that are unicellular and are able to make their own food, we call them as, we refer to them as autotrophic. So we are saying autotrophic are organisms which are able to make their own food. And those organisms, or those unicellular organisms that are maybe animal-like and are not able to make their own food because they lack that green coloring matter within them, then they depend on external assistance to get food. Then we refer to such organisms as heterotrophic. So heterotrophic are organisms which depend on external sources to get their food. They depend on external sources to get their food. And when we are talking about this unicellular organism like the protist, we say They are minute. We have close to 0.1 millimeters, 0.1 millimeters when we are talking in terms of size. How big are these organisms? They are close to 0.1 milliliters and in size, and some examples, they include our friend the Amoeba, we have the Euglena, we have the Parameciums, and so forth. They live in water, a place that is moist environment. They live in water and moist environment, and they differ in the locomotion. In their movement, they move in different ways. Some have whip-like tail, like the Euglena. They use their whip-like tail, the Euglena, to move. Others have a fine, hairy structure, like the Paramecium. They use that fair, hairy structure to move. And some of these unicellular animals, they do not harm human beings. Some of them are beneficials, others are not. For instance, those who are beneficials, we are told they are located within the digestive system. And if they are located within the digestive system, like the bacteria, then they help in digestion and so forth. Now, we are saying, if we use an example of a bacteria, we are told bacteria are about 1,000 times smaller than unicellular protist. And they are common. They are found everywhere in the environment. When you're talking about bacteria, we say bacteria are present in the air that we breathe. We have bacteria that are present there. And we are saying they are also present in dead plants and animals. Some live in digestive tracts and help in the digestion, some of these bacteria, and others live on the root nodules of plant, and they help in converting the nitrogen into state which can be absorbed by the plants. So they are found on the root nodules of plant. They change the nitrogen into a form that can be absorbed by the plants, and the nitrogen becomes fertilizer to these plants so that the plants can grow well. So we are saying some of the bacteria are not beneficials. Some bacteria are not beneficials. They cause diseases. For instance, we have diseases which are caused by these bacteria. And we're saying bacteria are found in everywhere. They are found everywhere in billions. Even they are found within the skin, on the skin. Some few questions before we embark on what we were to do today. One question is, if you are asked what is autotrophic, what is it? We had said that autotrophic, these are organisms which are able to make their food. And a question like, what is unicellular? We had said unicellular. These are organisms that are made up of a single cell. Now, when we're dealing with the unicellular organisms, we are saying that these unicellular organisms, they are made up of one cell only. That's why we call them unicellular. And these organisms have they are made in such a way they have a large surface area, so that compared to their volume ratio, they have a large surface area, and they rely on simple division for them to get their nutrients. They rely on simple division to meet their needs. An example of unicellular animals, we had given an example of an amoeba, and we said amoeba, it's a single cell, and it can be able to carry out all the 7 processes that are important for its survival inside that small cell. And what does the amoeba do? We have said the amoeba is located in ponds. Yes, they are there, amoeba, and they move by changing their shape. The amoebas have what we call pseudopodia. The pseudopodia, these are elongated, pseudopodia. Pseudopodia, this one, they will produce, depending on where the amoeba wants to move to, this pseudopodia will be able to formulate, and it will move the rest of the body. It will follow the same system. So we say an amoeba feeds on small organisms such as a bacteria. The pseudopodia, what will do, it will flow around the food, engulf it, and The food then is taken up in the cytoplasm, and in the cytoplasm, it is found, cytoplasm as an area called the cell vacuole, where the food will be located. The enzymes then will be released. It will be able to digest that particular food into small components that can be able to be digested. Useful substances such as oxygen will be able to diffuse, to pass through the cell membrane. Useful substances such as oxygen will pass into the amoeba. At the same time, water will also diffuse. Then waste substances like carbon dioxide will diffuse out, will go out through the same diffusion. So we find that the cell membrane of an amoeba is made in such a way that water can enter through, and it can also leave depending. So useful substances will be able to diffuse, it will take oxygen and release at the same time the carbon dioxide, release it out. When we have just touched on unicellular animals made up of one cell only, what about multicellular? This is what we can talk about multicellular animals. We say multicellular organisms are those organisms that are made up of many cells. A number of cells combine to form these organisms. And we can give examples of animals, even human beings are multicellular organisms. And we are saying multicellular organisms are more complex because these small cells will unite to make up tissues, and tissues will formulate system. And the system later on will actually make up the whole living organism. This is because cells of these organisms, some of cells of this organism are specialized for particular purposes only, and they play specific role and specific functions within the body of an animal. For instance, we have cells such as nerve cells that are made in a specific way. We touch on it to transfer the electric nerves from one end of the body to another. We also have the muscle cells. We also have, they perform different functions. We had seen that. When we give an example of a plant, we have the root hair cells. The way they are made in a specific way. They can be able to absorb nutrients, and they have large surface area for diffusion to take place quite well when we're talking about the root cells. Then we are saying we have the leafy cells that has a lot of stomata, so that it can be able to carry out the exercise of photosynthesis well. Now, today, we have been studying about cells, living organisms. We have said unicellular organism and multicellular organism are made up of cells. Today, we are going to learn about cell divisions, how the cells divides itself, cell division. When we talk of cell division, is the process by which the parent cell divides into two or more daughter cells. We are saying, what is cell division? We are saying cell division is the process by which the parent cell, is the process which the parent cell is the process by which the parent cell divides into two or more. Divides into two or more daughter cells. So this process of cell dividing itself into two or more daughter cells is what we refer to as cell divisions. Cell division usually occurs when part of the large cell, it occurs in part of the large cell cycle. In large cell cycle, the cell divides itself. We have a process by which the cell divides itself. It depends. There are majorly two processes. One process, we call it mitosis, another one is meiosis. But today, we shall deal with mitosis. what is mitosis and how mitosis takes place. But before we come to mitosis, we can say there are two distinct types of cell division. One is vegetative division, whereby each daughter cell is genetically identical to the parent cell. We call that one vegetative division. Vegetative division is whereby each daughter cell is genetically identical to the parent cell. And in such a case, we call it mitosis. Mitosis is whereby the cell will subdivide into two, and the two cells that are going to be formed are exactly like the parent cell. Such a kind of division is what we are referring to as vegetative division. Then we are saying the second process of division, we call it reproductive cell division. And in reproductive cell division is whereby a number of chromosomes in daughter cells is reduced to half to produce haploid or gametes, and this process, we call it meiosis. Meiosis is where the chromosomes that are found within the cells that are halved, meaning that it's just a half number that divide themselves into two that are halved, and we are saying they produce haploid or gametes. That's meiosis. Meiosis results into 4 haploids daughter cells by undergoing a round of DNA replication that falls into divisions. Now, we had said that today we shall concentrate on mitosis, not meiosis, and mitosis has phases. Maybe we are going to deal with the mitosis phases. Now, the phases of mitosis we want to see, and we had said that mitosis is a type of cell division whereby the nucleus divide into 2 daughter nuclei. We are saying mitosis is a type of cell division where the nucleus divide into 2 nuclei. So we said that in mitosis, the cell will divide into 2 identical daughter cells. They look like the parent cell, the parent cell. Then we are saying mitosis has got close to 4 phases. Okay, there are some books will say 5 phases, but the first one we normally call it interphase. The process before the mitosis steps in, that is interphase, when the cell is preparing itself for mitosis, that is division. So the correct phases that are there, that we can see, they are about 4 phases. And when these 4 phases are taking place, it is either the 3rd or the 4th phase. There is another process that steps in called cytokinesis. Cytokinesis. Cytokinesis, this is a process that steps in when during the 3rd or 4th phases of mitosis. And we are saying cytokinesis is the division of cytoplasm. Cytokinesis is the division of cytoplasm. Cytoplasm after the nucleus has already subdivided itself is the division of the cytoplasm immediately following the mitosis to form 2 following mitosis to form 2 daughter cells. So we had said we'd said cytokinesis is a process that steps in the third or the fourth stages of mitosis. Cytokinesis steps in the third or the fourth stage of mitosis. And we have said it is the division of cytoplasm after the nucleus have divided themselves. Then it is up to the cell cytoplasm also to subdivide itself. So when these cytoplasm is subdividing itself is when we say that is cytokinesis. It subdivides itself to cause two independent daughter cells from a single cell. So we are saying mitosis consists of four stages, basically consists of four basic phases. The first phase is called prophase. We have prophase as the first stage. Then the second stage we have metaphase. That is being the second stage. Then the third stage we call it Anaphase. Then the fourth and last stage is called Telophase. So when we are talking about mitosis, and we say mitosis is a process by which the cell is able to subdivide itself into two daughter cells that are exactly identical with their parent cell. So we are saying this process of subdividing itself, the cell will have to undergo the four phases. Maybe we can start with the prophase, the first stage. Prophase stage, we see how that takes place. On the prophase stage, we can say the chromatin, we have known the chromosomes. The chromatin condenses into discrete chromosomes. The chromatin condenses into discrete chromosomes. The nuclear envelope breaks down and the spindles form an opposite poles of the cell. I repeat, I'm saying on the prophase, what happens is the chromatin condenses within the cell, condenses into discrete chromosomes. The nuclear envelope breaks down and the spindles form opposite poles of the cell. We are saying the prophase is the first stage in mitotic process. Mitotic process is just the same process that mitosis has to undergo through. So we're saying there are important changes that take place during this prophase, and some of the changes that occur in the prophase are: one, we can enumerate these changes that take place during the prophase stage. One is the chromatin fibers become coiled. The chromatin fibers become coiled. They become coiled into chromosomes. They become coiled into chromosomes, which with each chromosomes having two chromatids. Having two chromatids. Yes, with each chromosomes having two chromatids that are joined. That are joined on the centromere. Now, that is just first part of the prophase stage. And we're saying on the same stage, the mitotic spindle composed of microtubes. So we are saying the second process that takes place within the prophase, we are saying the mitotic spindle, the mitotic, yes, the mitotic spindle composed of microbules, and proteins. Composed of microbules and proteins forms in the cytoplasms. So these spindles, they form themselves within the cytoplasm. That becomes the second process within the prophase phase, the prophase stage. Then the third is, in the case of animal cells, we are talking about plant cells and animal cells. What about if in the case of animal cells, the mitotic spindle appears a structure called asters, which surrounds each centriole pair. So in a case, maybe that cell that is dividing itself is an animal cell. Then the fourth process under stage 1 of prophase, we are told there are the two pair of centrioles move from each other towards the opposite ends of the cell due to the lengthening of the microbules that form between them. So we can find that the two centrioles move from one another towards the opposite end of the cell. Then on the fifth process, we are told the nuclear envelopes breaks up. Then the sixth process within the first stage, that is prophase, the chromosomes begin to migrate towards the cell center. That's another process that happens within the prophase. We find that the chromosomes will now begin to migrate towards the center. What we are trying, these all processes are under the prophase stage. Let us go to metaphase. Now, in cell division mitosis, cell division, metaphase happens to be the second stage that in meiosis process. We are saying metaphase, and for you to see, if you are talking about the plant cell, you can see for the root to subdivide itself, the cells that are found within the roots of a particular plant, they know how they subdivide themselves. How do they carry on the process? We are saying this onion root-like tip of that plant, the cell is in metaphase or mitosis when it replicates. It replicates the chromosomes or chromatids. It can replicate the chromosomes or chromatids that are aligned upon the equator of the cell and are attached to the spindle fiber. So the spindle fully develops into chromosomes. So we are saying there are important changes that take place within this metaphase stage. One of the changes that is not naming is the nuclear membrane disappears completely. The nuclear membrane disappears completely because we have the DNA and the RNA. So when the RNA disappears, then the DNA will not be there for a while, just for a while. Later on, it will reappear. So we are told the nuclear membrane disappears completely in the second phase of metaphase. So the nuclear membrane will disappear. Then after disappearing, if the cell was an animal, we are talking about in the case of if it was a plant, if this plant cell, if it was an animal cell, then two pairs of centrioles will align opposite poles of the cell. Then the second process that will take place within this metaphase stage, we have the polar fibers will continue to extend from poles to the center of the cell. So we are saying here the polar fibers will continue. So the polar fibers will continue to extend from the poles to the center of the cell. So they will be lengthened. Then we are saying, then after this process, we shall see that process that will take under this stage number 2 metaphase. What is going to transpire on the third process? We are seeing the chromosomes move randomly until they are attached to polar fibers. The chromosomes will move randomly within the cell. They will move randomly within the cell until they attach themselves to polar fibers. Yes, they will move. That's what's going to happen. They will move randomly within the cytoplasm until they attach themselves on the polar fibers. That becomes the third process under the second stage, the metaphase. Then the fourth, what will happen the fourth, is that the chromosomes are held at metaphase plate. There is a metaphase plate that are going to hold them. The chromosomes held at metaphase plate by equal forces of polar fibers, pushing the centromeres of the chromosomes. We had said in the fourth stage of this metaphase, we had said in the fourth stage of metaphase, we have chromosomes that are held at the metaphase plate. We have chromosomes that are held at a metaphase plate. Now, by equal forces, they're held by equal forces of polar fibers, pushing centromeres. They push this, what you call centromeres. Centromeres. They move the centromeres of the chromosomes. That becomes the fourth process under the metaphase. Then after the metaphase, we go to that stage of mitosis, and that stage is called anaphase. This is the third stage of mitosis, and in this third phase of mitosis, what happens? Now, the paired chromosomes, or the sister chromatids, separate. They separate and begin moving towards opposite ends of the cell. We are saying in this anaphase stage, the paired chromosomes, or the sister chromatids, they separate and begin moving to the opposite end of the cell. Changes that occur in anaphase. What changes occur here? The changes that occur in anaphase, the first change is the centromeres. The first change is the centromeres begin to move apart. That is what observed, that's what explains. The centromeres begin to move apart. Then two, once they separate, the second they separate, once they separate, once once they separate from each other, from each other, it is considered each other there is considered chromosome. Once they separate from each other, each individual becomes full chromosome. Each becomes full chromosomes. They are referred to as daughter chromosomes. They are referred to as daughter chromosomes, completely identical to the first parent. They are almost identical to the first parent, cell. Now, when we leave this stage anaphase, we go to the fourth stage, which is called telophase. And on the telophase, it is the fourth stage of mitosis, the telophase. On the telophase, the cell is nearly done. That cell, it is nearly done dividing, and it starts to re-establish its normal structure. Here we find that the cell division is almost done. Almost done. And it starts to establish its structure. It starts to establish structure. The mitotic spindle is broken down into building blocks. Two new nuclei form, one for each set of the chromosomes. So you find that there's a time when the nuclei subdivided, it had disappeared because there was no RNA, so the DNA disappeared, but right now the nuclei now is coming back. So we are saying the two nuclei form one for each set of the chromosomes. The nuclear membrane and the nucleo disappear. The nuclear membrane and the nucleo disappear. Then the chromosomes begin to decondense and return to its stingy form. All the above phases occur in sequential order. And we had said that when the stage of anaphase and telophase is taking place, there is a stage called cytokinesis that peaks on, that starts immediately under stage number 3, that is anaphase and telophase. There's a process called cytokinesis that takes place, which is a process that takes in it is a process by which the dividing cell content, they make two new cells, and it starts in anaphase or telophase. So when these two processes, the third and fourth processes taking place, another fourth process called cytokinesis takes stage, and it ensures that there are two daughter cells that are completely a replica to their first parent cell. So we are saying stages in mitosis. The first stage is prophase, then the second stage is metaphase, the third stage is anaphase, and the fourth stage is telophase. We are saying before the first stage comes in, that is the prophase, there is another stage we called interphase. Interphase, the process of mitosis had not yet started, but the cell is just readying. The cell is readying itself. It's preparing itself for mitosis. So there is that stage they call interphase. Then the first stage, prophase. From prophase, we go to metaphase, anaphase, and then telophase becomes the last stage. Now, we are asking ourselves, mitosis as a process, we have said it's a cell division whereby a cell is able to divide itself into two. Does it have any importance? what is the importance of mitosis? The importance of mitosis, we had said mitosis is a cell division. It is also, the mitotic cell division is also called mitosis, or one cell dividing into two. Plants can use mitosis to reproduce in a process called rooting. So if you look at the roots of a plant, they don't remain stagnant, they grow. So when they are growing, we call that process rooting, meaning the roots of that particular plant is elongating. The roots can only elongate under the process called rooting when a process called mitosis is being carried on, because the cells are multiplying. How are they increasing? How are they multiplying? They are multiplying when a parent cell is subdividing itself into two. So we are saying one of the importance use of mitosis in plants, plants are able to carry out a process called rooting. And in this process called rooting, the plant is able to develop roots, these roots that can be able now to absorb nutrients within the soil. Rooting process. So for the roots to develop, for the cells of that particular roots to increase, it will only increase through a process called mitosis. So mitosis assists the plant to ensure that it has enough, the roots grow and are able to absorb the nutrients from the soil. That's one when you're talking about the plants. In also multicellular organisms, mitosis is used to repair small wounds and even large wounds. If an animal has a wound, then for that wound to be repaired, mitosis process will take place to replace the dead cells or the cells that are worn out. They will only be replaced through a process called mitosis. Also, mitosis, it is another, it helps if the multicellular animals are growing. They can only grow under the process called mitosis because it causes growth. The cells will be able to subdivide into other cells that are similar. So we are saying growth and development from a single cell. The multicellular animals, they were once a single cell, but growing from that single cell, there was cell division, cell subdivision. The single cell kept on dividing itself into other cells, and therefore that particular organism was able to grow. So growth and development into full organisms is acquired or is made through the process called mitosis. Then we are saying is okay. In unicellular animals, how is mitosis important in unicellular animal organisms, such as the bacteria? The bacteria, through mitosis, it is able to carry out a process which is similar to asexual reproduction, making identical copies of that single cell. Therefore, if we have a single cell of a bacteria, for you to have another single cell of bacteria, then the process of mitosis is important so that another single cell of bacteria is created. It is only created through cell subdivision using the process of mitosis. Now, in multicellular organisms, we had said mitosis produces more cells for growth and repair in multicellular organisms. Then in unicellular organisms, which have got only one cell, it is able to replicate, to produce another similar cell by mitosis. There are a number of questions we can ask ourselves on mitosis. One, we can now ask ourselves, what is the function of mitochondria? What is the function What is the function of mitochondria within a cell? To respond to such a question, we had said mitochondria is a substance that is located within the cytoplasm. And we had said it is only under the help and assistance of an electron microscope to be able to determine what functions mitochondria play. Okay, with a light microscope that has enough lens, you can be able to see a mitochondrion, but you cannot be able to see the functions. But under electron microscope, it has been established that mitochondria serves as a powerhouse. It is able to provide energy for different processes that take place within a cell. Therefore, to respond to this question, what is the function of mitochondria within a cell? We say mitochondria serves, it is able to provide energy for different processes that are carried out within the cell. So mitochondria is there to supply energy. It has a way of trapping energy and making sure that that energy is in the state that is able or that can be used by the different functions that take place within the cell. What part, a second question we can have, what part of a cell that controls substance that enter and leave the cell? What part of a cell So what part of a cell that controls substance entering and leaving the cell? Now, if you look at a cell closely, we say the substance that can be able to allow the part of a cell that allows some substance to enter and leave the cell. This is a simple question that depends on how you have read and it depends on your memory. So we have said that the part of a cell that controls substance entering and leaving the cell is the cell membrane. And we had said cell membrane is semi-permeable, meaning it allows only substances that are beneficial to the cell and is able to eject out. It is able to reject substances that now are toxic, that have been the byproduct of the process that has taken place within the cell. So some substances can be allowed in like oxygen, then substances like maybe carbon dioxide can be let out. So we are saying cell membrane is made in such a way that it can allow useful substances within the cell and leave out the byproduct of the processes that has taken place within the cell. So that part of cell that controls substances is called cell membrane. The question we have of definition, what is mitosis? What is mitosis? And I believe mitosis is what we have been dealing with. We said mitosis is part of the cell division. We said there are two cell divisions. We have mitosis and meiosis. And today we are looking about mitosis. We said mitosis is one cell dividing into two. One cell dividing into two. And once it divides itself into two, then that is, and these two producing two other daughter cells that are similar to the parent cell. That is in a simple way we have defined what mitosis is. We can have last question here. List three ways in which mitosis is important. list 3 ways in which mitosis is important. This part can be answered with the last part of the topic when we were dealing with the importance of mitosis. We had said some of the importance of mitosis. One, we had said single-celled organisms such as bacteria are able to make identical copies of a cell. So through mitosis, single-cell animal like bacteria are able to multiply themselves. They are able to replicate. They add to their number. That's one. Then another advantage, we talked about multicellular organism. And on multicellular organism, we had said through mitosis, these multicellular organisms are able to produce more cells for growth, then more cells for repair, and then also more cells to repair maybe small wounds and big wounds are repaired through the process of mitosis. So we have seen that multicellular organisms are able to repair themselves to grow through this process of mitosis. We had seen in the cell division, we had seen that we have 2 types of cell division. We had seen the mitosis cell division, and we had also seen meiosis. So we said in cell division, today we are going to deal with the mitosis, and we had said that with the mitosis, it is a situation whereby one cell divides itself and produces 2 cells, 2 daughter cells. So when one cell is able to divide itself and produce 2 identical cells, then we call that process cell division. And we said that we call that process mitosis. And we said this process of mitosis has 4 phases. We had looked the first phase being the prophase. We had seen the prophase, and we had seen the processes that are associated with the prophase as phase or stage. And from the process stage, we had seen also other stages, the metaphase. We had seen also, we have the metaphase. That being the second process in the mitosis. And then we have the anaphase. And then lastly, we had the telophase. So when we were talking about mitosis and all these phases that the cell has to undergo before it divides itself into 2 daughter cells that are identical to their parent cell, we said for us to understand this mitosis properly, we need to observe, at least to have a diagram that is showing how mitosis takes place. So first, we know the cell at its normal phase, it can appear like so, saying maybe this a cell before cell division, it is existing in such a manner. In between, we have the nucleus, but inside the nucleus here, we can have the chromosomes, which have some DNA and RNA that are located within the nucleus. And nucleus is there to dictate the genetic characteristic that a particular organism is supposed to have. Therefore, let us try to explain this process of mitosis. Now, if this is a cell at its initial stage, others can say maybe that's interphase, but it's a stage before the cell starts cell division. We can have this kind of a cell. Now, when you look within, that's what we can call the chromosomes, that the cell is just still at its initial stage, but there are a lot of activities that are being replicated. So we can say this is a normal life. This is a normal life stage of a cell. This is a normal life in a stage of a cell. So I think that's a normal life, life of a stage in a cell, when the cell is not dividing. When the cell is not dividing. So once it starts to divide, then we shall see some differences. Here we see it is when genetic information is being replicated. So what happens here, we are saying this is thus the first stage, and we are seeing that the genetic information is being duplicated and the cell's activity is high. The cell's activity is high. So we can refer to this one as maybe 1. But when we go to the same mitosis, what is happening in another phase, then we can see that changes can occur where you find that now the chromatids within the cell, they line up along the center of the cell. So we can have such a structure. Yes, we can have such a structure. Then you find now these chromatids are lined up. So you find that they are lined up within the cell, but the tension is there. Yes, the tensions are there. Yes, here we see the chromatids are lined up along the center. They are lined up along the center. So you can see along the center how the chromatids are lined up. Now, this is when we say the process of mitosis is on. It will go to the extent whereby these chromosomes will subdivide themselves into halves, and later on they will form their own cell, daughter cells that are exactly a replica of their parent cell. We can have another diagram here showing that process of what takes place. Now here we can have such structure. Yes, we can have such structure, and so we can find the tensions are there. So from this you can find that the forces that were there, the tensions that were there were pulling the chromosomes towards the end of the poles within the same cell. They have managed to pull these chromosomes in such a manner. So we find that the daughter chromosomes separate and move towards opposite end of the cell. Here are the daughter chromosomes separate and pull towards the end of the pull towards the end of the cell. by the spindle fiber. So you can see the forces, the spindle fiber, they have managed to separate these chromosomes into halves, and they're being pulled towards the end of the cell at the polar sides of that particular cell. Then we can advance ahead and see what the fourth process that takes place in the mitosis. And in the fourth place, whatever takes place, we can find that we shall have such a shape. It is still the same cell, but something has happened. You can see something has been created in between there, meaning at least the cell division is on. Mitosis is on, and it's almost managing to subdivide the same cell into two. So the chromosomes become visible. The chromosomes become visible. They become visible now. The chromosomes become visible, and two chromatids which are connected to each other at the point known as centromere. The chromosomes become visible, and two visible and two daughter chromatids. So we find the two daughter chromatids which are connected to each other. Which are connected to each other at the point known as centromere. When you say at the point known as centromere, it is this particular place where they are still connected. But we are seeing how the two daughter chromatids are coming up in this for the process of mitosis. Then we find, as it goes ahead, we find another process altogether. The daughter chromosomes again. My drawing is not to scale, but the two cells are supposed to be replica. They are supposed to be identical to each other. The daughter chromosomes here the daughter chromosomes gather at the end. They gather at the end of the cell The spindle fiber, the spindle breaks down. The spindle breaks down, and the two daughter nuclei with the identical chromosomes to each other. With the two daughter nuclei with identical chromosomes. Yes, they have identical chromosomes at each other. So you find that these, at the end of it, we find that now we have two daughter, two cells that are identical to each other, and they are just a replica to the parent cell. The whole this you can find now we have two daughter cells that are similar to the first parent cell that started this mitosis, that is cell division. This diagram is important to explain the phases of cell division, that is mitosis, from the prophase from the first stage, that is prophase, the metaphase, the anaphase, and the telophase. Now, when we are talking about cells, we say cells are minute, but they do unite. And when many cells unite together, they form what we call a tissue. We have tissues. Many cells make up tissues. So tissues and many tissues, when the tissues of the same grouping come together, they formulate what we call organs. Then from many organs that are together, you find they're working together, then they bring up systems. Maybe we shall start. We all know that in order to grow, we need to develop from a cell. Growth can only happen when there is development in a cell. So growth and development from a single cell, along the way, this single cell will again continue dividing itself, and as it goes on to divide itself into daughter cells, and the daughter cells goes ahead to subdivide themselves again into so other many different types of cells they form. From each of these different types of cells they form, then we shall have these large bodies being formulated all together. What we are saying, when cells of the same type group together, then a tissue is formed. And such a tissues that are formed, we can give example of muscle tissue. When you take a cell, a muscle cell, when muscle cell combines, it will form a muscle tissue. Then from the muscle tissues, for example, we have muscle tissue. We can also have the fatty tissue. We can have also bone tissue, meaning there are many cells that have combined, many bone cells have combined together to form a bone tissue. And again, from bone tissue, we can have blood tissues, meaning blood tissues. There are a number of cells, whether it's red blood cells, white blood cells, they have formulated what we call blood tissue. Then we have the skin. The skin is made up of many cells, and then they formulate what we call the skin tissue. And then even, we are not talking about maybe animals alone, even plants. When plants come up, small cells come up, they can formulate what we call the bark tissue. We have talked of skin tissue, that's human being, but if it's a tree or something like that, then we can get what we call a bark tissue, meaning it is a number of cells have combined to form a bark tissue. Yes, a bark tissue. So we are saying different tissues group and work together. Different tissues can group themselves together and work together to form organs. So when you group many tissues together, then you will form what we call organs. Now, these organs, which also have specific jobs to do, organs in our bodies, example of organs in our bodies, if we're talking about human beings or animals, then organs in our bodies, we have an organ like the heart organ. Yes, we have the heart organ. The heart, we have the kidney. So when we talk of organs example, we have the heart, we have the kidney. We know the kidney is responsible for cleaning, a lot of cleaning of the blood and so forth. So we have the kidney as one of the organs. Apart from the kidney, we have the lungs. We have the lungs. We have the skin. The skin that we have is an organ. The lungs that we have that helps in breathing system, it's an organ. And then we have the brain. Brain is an organ. Brain is a combination of many cells and tissues. So we have the brain organ, and we also have the stomach. Stomach is another example of an organ. You have also the stomach. So we are saying, if we go to an example of plants, maybe in plants we have organs such as their roots. So roots that are located within the trees, we have roots that are organ. They help in absorbing nutrients from the soil. So we have the roots, we have the stem organ. The stem is responsible for translocation of water from one part of the plant to another. So we have the stem, then we have the flowers, it's an organ, and then we have the leaves. They perform particular functions that are important. Now, in humans and other higher animals and other animals, there are different types of organs that are grouped together or they work together. So when different types of organs work together, then whatever function they're performing, they become a system. So when different organs group together and they work together, they form systems. An example of systems in human beings, we have the circulatory system, and we have the skeletal system that we know it is important in supporting a particular living organism in terms of standing and movement. We have the respiratory system that assist in terms of respiration. And we have the digestive system. Digestive system, it has a combination of many organs that play important role in digestive, where the digestive is concerned. We shall see, we even have a reproductive system. So in our next lesson, we shall deal with this system. We shall check their functions. What are their functions? If we talk of reproductive system, what are their functions? If we talk of the nervous system, what are the functions? What does nervous system do? If we talk of endocrine system, what is the role of this endocrine system? Even when we talk of excretory system, how does it function there? What organs combine to form this excretory system? That is the end of our lesson today. Thank you.